아, 이름 예쁘다.
나 그냥 한국말 못하는 줄 알고. 이 색깔.
한번 만나요. 그래요? 정말요? 정말? 언니 근데 진짜 아니 진짜 어려웠어요. 나 깜짝 놀랐어. 나 진짜 놀랐어. 수빈. 진짜 완전
Okay, we're ready to divide. Okay, so leading up to where we're at here, by this point in time, MPF is pretty much done a lot of what it has to do. As we talked about on Friday, we've gotten rid of the nuclear membrane now. The lamins are phosphorylated, so the nuclear envelope's gone. Chromosomes are condensed. They're all ready to be segregated. So now all we have to do is make the mitotic spindle, segregate the chromosomes, and we're done. Okay? So that's where we're at right now. So the first thing that you're going to see, if you take a look at the slide that's up there now, is that there's going to be very specialized microtubular functions in the mitotic spindle. Okay? Now, the thing to understand is that these are operational distinctions. Okay? The microtubules that are involved here are all made out of the same stuff. Okay? So there's nothing predetermined that you can go in and say that a polar microtubule is any different than a kinetochore microtubule or an astral. It just happens to say, okay, this is what you wound up doing during this particular round of mitosis. I mean, tubulin's tubulin. It does not know what the microtubule it gets incorporated into is going to do. Okay? So, but just to take a look at the division of labor here, it's actually pretty straightforward. Remember, all of the microtubules originate within the centrosome or the M-top. By this particular time, going into mitosis, the M-top is duplicated. We'll get to that right on the next slide. So the centrosomes have duplicated, and they've migrated to opposite sides of the cell. And that migration is what determines the axis of cell division. Okay? The chromosomes don't have anything to do with it. They're passive players in this. But what it means is that all of the microtubules that are coming out of each of the two spindle poles are radiating with their plus ends going out. Okay? So if we take a look at it, only a very, very small subset of the microtubules that come from the pole actually capture or attach to the sister chromatids. And those are referred to as the kinetochore microtubules. Okay? They're the only ones that directly contact and hook up with the chromosomes themselves. All right? And the reason is that that's a really small target. It's a really small bullseye on a chromosome. Now, our chromosomes are relatively large. Our kinetochores are large, and they have binding sites for multiple microtubules. In the case of the yeast, Saccharomyces cerevisiae, that kinetochore binds one microtubule. Okay, so if you're looking at segregation of chromosomes in yeast, they move simply because of one microtubule attached to each sister chromatid. So that organism is really living on the edge. Okay? So the kinetochore microtubules attach here, and basically they are actually providing the direct track for the chromosomes once they segregate, is that this sister chromatid is going to be moving down that kinetochore microtubule towards the pole, and what you're going to see is the kinetochore microtubule depolymerizes behind it. And that's going to be anaphase A. So the chromosomes move towards the spindle poles. And it's going to involve microtubule depolymerization. You're also going to see down here, it's one of the few times that you see minus N depolymerization taking place. So just think anaphase A shrinkage. Okay? Now, we've also got these microtubules that radiate out towards the plasma membrane. And they have a star-like appearance, and because of that, they're referred to as astral microtubules. Because of that, it's a starburst. They are going to attach the plus ends of the astral microtubules attached to the plasma membrane. And what they're doing is that they're anchoring the two spindle poles in place so they're not moving around and wobbling and everything. It gives them a fixed attachment point. And what's going to happen there is that you're going to see there are dinings that are going to be anchored to the plasma membrane. Now remember, dyneins are minus N motors. They move towards the minus N. And what you're going to see is one of the movements then during anaphase B happens at the same time as A. So while the chromosomes are moving towards the poles are actually going to be moving further apart. And one of the ways that we're going to do that, it's very simple, is that these dyneins that are anchored out here to the plasma membrane are going to be grabbing a hold of these astral microtubules and pulling them. So the poles are actually getting pulled back towards the plasma membrane. So this one's going to move that way, this one moves that way. So they're actually moving apart. And just remember, at the same time that's happening, the chromosomes are moving towards the poles. So everything's trying to move further and further apart before cytokinesis. Then finally, we've got these leftover microtubules that literally don't have anything better to do, so they become polar microtubules. These are the guys that were not lucky enough to spew out and attach to the plasma membrane as astrals. They're the ones that didn't have good enough accuracy to capture kinetochores. So the cell goes, all right, we got leftover tubulin, let's use it for something. The polar microtubules overlap. And here's where you're going to see these bipolar kinesins that are going to be walking towards the plus ends of the polar microtubules here. And as they do so, you're going to have a kinesin up here that's walking this way, and it's going to shove the overlapping microtubule above it back that way. And similarly, that same kinesin that's walking towards the plus end of this one is taking this microtubule below it and pushing it back that way. So the way to think about it, the poles are being pulled apart by the astral microtubules, and they're being pushed apart by the kinesins that are on the polar microtubules. So anaphase B really comes down to just moving the poles further apart. Anaphase A is very simple. The chromosomes migrate towards the poles. Okay? So, in a nutshell, that's pretty much everything we're going to be looking at. All right. First thing we got to do, though, is duplicate the centrosome. Okay? Remember, the centrosome has a pair of centrioles. They're RNA protein constituents. There's no evidence of DNA being in there. But yet, centrioles duplicate semi-conservatively, just like DNA. And in fact, centriole duplication, or centrosome duplication, takes place during S phase, just when your DNA replication takes place. All right? So what happens is that you have a pair, centrioles, they always lie perpendicular to each other in the centrosome. No one really knows why. They separate, and now what happens is each one of those original centrioles serves as a template to cause the duplication of a new daughter. So again, we've got mothers and daughters going on here. And so what it means is each centrosome now contains one original centriole, and it's got a new one. Okay? It's a very poorly understood mechanism. If you want to look at a neat problem in cell biology to go solve, figure out how centriole duplication takes place. If you want an even neater problem, keep working on it, and now figure out how these duplicated centrosomes know where they're supposed to go. You know? I mean, in this case, basically, these two centrosomes migrated. One went up to the top, 12 o'clock. The other went to the bottom, 6 o'clock. And that would mean this cell is going to divide along this axis. Okay? But you could also rotate this 90 degrees, and in some cells, they will migrate to 3 o'clock and 9 o'clock, and the cell is going to divide vertically. So again, we have very, very little understanding or appreciation of how the centrosomes know where the heck they're supposed to go. But they're determining the axis of cell division, and again, the chromosomes play absolutely no role in this. You can, in fact, pull the chromosomes out of the cell, and the centrosomes will still migrate to where they're supposed to go. Okay? The key thing here is it happens during S phase, just like DNA replication, and it's when this process is completed that then and only then is CDC25 now active. Okay? So that phosphatase is going to reverse the Wee1 inhibition of MPF. That phosphatase becomes active once the centrosomes have duplicated. That's what it's looking for. Now remember, it can't do anything until CAC has phosphorylated threonine 161. And CAC does not do that until we've completed G2. So centrosome duplication takes place first. Activation of CAC only takes place after the ribosome number is doubled and the cell has doubled in size. Okay? So you've got a cell that's now twice as big. We've duplicated the centrosomes. The nuclear membrane is gone. So now what we have to do is to start the process of what's called kinetochore capture. And the idea is that each of those two sister chromatids has to be captured and all of the microtubule binding sites on the kinetochore occupied by opposite spindle poles in order to get the two sister chromatids to segregate to each of the two daughter cells. Okay, so the first thing that we're going to do is that remember that microtubules in a non-neuronal cell are already pretty unstable. We're going to make them even more unstable. And here's another target for MPF. There's a protein called XMAP215. The function of XMAP215, it's a microtubule-associated protein, is it binds to the plus ends of microtubules, and it basically helps keep them relatively stable in a non-neuronal cell. It's going to be inactivated by phosphorylation by MPF. Okay, so when MPF becomes active, it's phosphorylating the lamins, it's phosphorylating histone H1, it's phosphorylating condensins, it also phosphorylates XMAP215, and that inactivates it. And now what happens is, here's where the other guy, kinesin 13, can now become more robust in its activity. And the way that it does that is, remember, kinesin 13 walks towards the plus ends of microtubules, and it kicks off the tubulin at the plus end. It makes them very unstable. And so the way to envision what's going on here is that at the spindle pole, it's going to be spewing out from each of the two centrosomes. They're going to be spewing out microtubules in every direction. Now, some of them that go in the opposite way from the chromosomes attached to the plasma membrane, they become astromicrotubules. They're stable. The others are spewing out towards the chromosomes. And they go out, and if they do not capture a kinetochore, think of the kinetochore as the ultimate in a plus-end binding protein. So a microtubule comes out, it's trying to capture a kinetochore, but it is a really tiny target for any given microtubule to hit. So most of them are going to miss it. And what kinesin 13 does, it allows that microtubule to very, very rapidly depolymerize. It goes all the way back, it depolymerizes, and the centrosome spews out another one. It's hit and miss. So in essence, what you're doing is you're speeding up the degradation or the depolymerization of your microtubules to try and take advantage of an inefficient process to allow the cell to have as many chances as it can get to ensure that the microtubules can capture their kinetochores on each of the two sister chromatids. So that's why the microtubule dynamics change so much when the cells go into mitosis. Now, when we get to metaphase, everything stops. Everything just sits there, and it's good to go. But until then, the microtubules are going to be very, very, very unstable. Okay, now we're switching gears. We're going from looking at the microtubules to the target. Each chromosome has a unique DNA sequence. So here's a pair of sister chromatids. So that means they just completed DNA replication. They're held together by the cohesins. Each one of the chromosomes then has a spot that you can see cytologically. It's a constricted region. So whenever you look at mitotic chromosomes, you always notice that every chromosome has this little constricted region. Sometimes it's in the middle, sometimes it's closer to one end or the other. That's the centromere. The centromere is the starting point to build a kinetochore. The centromere is a DNA sequence. Okay, so the starting point, and it's a non-coding region of DNA. Doesn't have a gene, doesn't encode a protein. It is a structural region of DNA. And if you take a look at the sequence up here, it's very unusual. This is the centromere from a yeast chromosome. Human centromeres are very, very similar. And you notice it's virtually all A's and T's. Very few G's and C's. So it's a very highly AT-rich region of DNA. Each chromosome has one and only one centromere. You don't ever want to have two. You don't want to have three. Because those are going to nucleate the formation of kinetochores. And if you have more than a single centromere on a chromosome, which you can genetically do in yeast, by the way, and you can engineer a yeast chromosome to where it has two centromeres, and it's now called a dicentric. Well, the problem is, imagine if we take this chromosome, and if we put another centromere down here. Well, what the problem with that is, there's a 50-50 chance that, let's say, this centromere gets captured by a spindle pole over here on the left. We put another centromere down here, it could be captured by the mitotic spindle pole over here on the right. What's going to happen to that chromosome when it goes into mitosis? It literally gets ripped in half, and it's lost. So that's why there's very strong selection to make sure you've only got one centromere per chromosome. Okay? But it's non-coding, it's structural. The centromere is a binding site then for a group of proteins that collectively form the kinetochore. So the kinetochore is made out of proteins. And the kinetochore is going to be the target that the microtubules are looking for. And again, the way to think about this, guys, is that the kinetochore is the ultimate in a plus-N binding protein. So once a microtubule, remember these guys were all spewing out of the centrosome over here. They're coming out. If they don't capture a binding site in the kinetochore, they depolymerize, we try it again, we try it again, we try it again. Ultimately, all of the binding sites in the kinetochore get captured by microtubules. And now they become all stable. Okay? So the challenge that we have is this. Here's a pair of sister chromatids. We want to get the sister on the right, and they're held together by the cohesins, so that's keeping them together until we get to anaphase. The challenge is we want the sister chromatid here on the right to just be captured by microtubules that come from the spindle pole over here. We want to get the sister chromatid here on the left to be captured and all of its kinetochore binding sites bound by microtubules that come from the left. Because we want this chromosome to go left, we want that one to go right. Well, the problem is the capture process doesn't work that way. That is, when these microtubules are coming out from the poles, they don't have a choice over which kinetochore they bind to. And so the system is sloppy. And the system has to be set up in such a way that if you don't succeed in doing this the very first time, how does the cell not go into anaphase until we have what are called a bipolar or an amphitelic attachment? And here's what way we're going to do it. Is it loaded into the kinetochore? We've got a kinase and a phosphatase. Aurora B is a downstream kinase that gets activated by MPF. So we've got a kinase and a phosphatase that are in the kinetochore. The kinase is going to be on the inside. The phosphatase is going to be on the outside. So here's a case where we're going to control proteins in the kinetochore being phosphorylated or dephosphorylated by tension. In essence, only when you form a bipolar spindle, like you see here, can you guys see that the tension is going to be equal and it's pulling the two sister chromatids? We're trying to pull them apart. Well, this is the only time where proteins that are in the kinetochore are literally being tugged in opposite directions with equal force. And that is going to be the signal that says your spindle is attached and it's good to go. And what you're going to see is, the way that it's done, is that when we pull those two sister chromatids apart this way, proteins in the kinetochore can no longer be phosphorylated because we've literally pulled them away from the kinase and we've pulled them into the phosphatase. So here's a unique case where what you're doing is that you're regulating phosphorylation and dephosphorylation by pulling the substrate away from the kinase and allowing it to go to the phosphatase. You're mechanically moving the substrates. Okay, so here's what we're dealing with. Okay? Let's take a look at the tension first. If we take a look over here, if we have absolutely no tension, so this would be a pair of sister chromatids. They're just hanging out and they're waiting to be attached by the microtubules. So that's the starting point. We're going into mitosis. None of the kinetochores have been captured yet. There's no tension on anybody. And what you see here is that these kinetochore proteins are basically being exposed to Aurora B kinase, which is on the inside. So kinetochore proteins, you don't have to worry about the names. The book goes into all sorts of gory detail about the names of the substrates. Don't worry about it. Just think of it that when you've got sister chromatids that are not under tension, kinetochore proteins are phosphorylated. Cool with that. Here's where we want to go. We want to go from here to where they're under equal and opposite tension. Okay? When we do that, you can see that the kinetochore proteins here in the middle are being pulled apart from each other. And they're being pulled away from the kinase, and now they're exposed to the phosphatase. So all of these kinetochore proteins are now dephosphorylated. And the only way they can all be dephosphorylated is if both sister chromatids are in this attachment. So now we can just move over here. This is going to be referred to as a bipolar or an amphitelic attachment. Amphitelic meaning both are attached. This is the end goal that we're going for. These are all intermediates that you go through in order to put this together. We don't want the cell to go into anaphase with any of these attachments here because bad things will happen. You will get missegregation of your chromosomes. But you can see the one thing that's in common to those three misshapen attachments, we've always got proteins phosphorylated because one of the two sister chromatids is not going to be under tension. And that's going to be the mitotic spindle checkpoint. Okay? So let's take a look at the first scenario. This is where we want to go. It's trial and error. So let's say we're trying to set up, and what happens here in the first round is that we have what's called a merotelic attachment. Get some cool words here. Merotelic means the following. The sister chromatid over here on the right got captured by microtubules coming from the pole over here on the right. Cool, that's what you want. Well, but it also got captured by microtubules coming from the left. Okay? The sister chromatid on the left is unattached. Okay? So now you've got a problem. We've got one sister chromatid that can't segregate. It's not attached to anybody. So it's going to distribute randomly. So see what would happen. If we went into anaphase here, the sister chromatid on the left is going to be drifting aimlessly. It means it's got a 50-50 chance of going with the daughter cell on the left or the daughter cell on the right. You don't want to bet your chromosomes on random outcome like that. What's worse is the sister chromatid on the right is attached to microtubules coming from the right and the left. What's going to happen to it if it's not? It's going to get ripped apart. So that's not good. Monotelic. Monotelic now, what this means here is that one of the chromosomes, this sister chromatid, is not attached at all. It's a free drift, 50-50. It means that the chromosome on the right did get attached. Okay? So what this means is that this guy over here is going to segregate to the right. This one is now random. Okay? But again, can you guys see? There's proteins that are still phosphorylated. And that's because we don't have equal tension pulling the kinetochores apart and into the phosphatase. Syntelic. Syntelic means both sister chromatids attach to microtubules from the same pole. So in a syntelic attachment, both sister chromatids are going to segregate to one of the two daughter cells. That means the chromosome is going to be lost from the other daughter cell. So what it means is one of the two daughters is going to acquire 2 copies of the chromosome instead of just 1. So that means it's going to be trisomic. The other daughter cell is going to lack that chromosome entirely. That's generally a lethal event. Okay? So here's what's happening. We go through all of these intermediates in order to form an amphiphilic attachment. And the way that that occurs is that all of these attachments are going to be unstable. And that is the microtubules that are bound to those kinetochores. As long as there are proteins phosphorylated by Aurora B kinase, the microtubules are going to depolymerize, and we're going to try it again. We try it again, and we try it again. In essence, so what you guys can see is chromosome capture is a random process. It's hit or miss, it's very inaccurate, and it takes a while for this to occur. That's why when you go into mitosis and you see all this movement of chromosomes and everything, trying to line up in the equator, it takes a while. This isn't fast. But the point is, this phosphorylation is what allows the process to get a do-over repeatedly until it happens. But this phosphorylation of these kinetochore proteins is what activates the mitotic spindle checkpoint. And that is what is keeping the cell from going into anaphase. Okay, now, assume you get captured. Someone just woke up. Okay. I'll leave a message for you. Okay. Okay, so can you guys sort of see what has to happen with capture? It's random, it's messy, it's sloppy. The cells don't want to divide until it's done right. Cool with that? Okay, now, here's what we've also got to do. Here's your chromosomes. The problem is we need to get them all in the middle. It's only when they're on the equator of the mitotic spindle are the forces exerted on those two sister chromatids exactly equal and cancel each other out. That is the only way that all of those kinetochore proteins can be dephosphorylated. Well, here's the problem. What do you do when we've got a pair of sister chromatids down here? And this sister chromatid got captured from the poles on the left. This sister got captured from the poles on the right. Okay? It's amphiphilic. But can you guys see that the force is not going to be the same on them because it's not centered in the middle? That's how most of the chromosome capturing takes place, is that the chromosomes aren't going to wind up right here in the middle to begin with. We've got to have a mechanism to scoot them over. So as you guys can see here, this sister chromatid pair is on the left. we need to move it to the right in order to get it on the equator. The way that this is going to happen is that we're going to have compensatory shrinkage and growth of the kinetochore microtubules while the chromosomes are still attached. So if we want to take this pair of sister chromatids and move them over to the middle, what we do is that this kinetochore microtubule coming from the right is going to depolymerize. Now you'll see in a minute that does not cause the chromosome to let go. The kinetochores are sort of holding on to the chromosome or the microtubule with a weight. And you'll see that in a second. So what happens is the plus end of this microtubule over here depolymerizes. And there's kinesin 13s that are stuck on the kinetochore that are causing the plus ends of these microtubules to let go. Okay? The chromosome remains attached. But what it means is this microtubule gets shorter. So now that means it can move towards the right. But we need to give it a shove. So at the same time that that is happening, this microtubule over here on the left actually polymerizes. And now we actually have a kinesin here that's shoving the sister chromatids towards the opposite pole. So in essence what we're doing, this microtubule, this kinetochore microtubule shrinks. And so the chromosome is able to slide down. It remains attached, but it slides down to the right. Literally, the microtubule on the left is getting longer, and it's pushing the chromosome that way. So this process of pulling and pushing is what's necessary to take all of these sister chromatids and to get them perfectly aligned right in the middle. Because only when they're equidistant from the two poles do all the forces cancel out. The kinetochores are now being pulled with equal strength from both sides, and all of the kinetochore proteins are dephosphorylated. That's what we're looking for here. Here's the checkpoint. The checkpoint requires the anaphase promoting complex. And you see all these X's? That's because don't worry about their names. We don't care. There's only one protein that matters here. This is the cohesin complex. It's holding our two sister chromatids together. When we go into anaphase, the anaphase promoting complex has to cleave one of these cohesin subunits, and it's the protein SEC1. That's all you need to know. Here's the anaphase promoting complex. The anaphase promoting complex has already ubiquitinated cyclin B. It's basically it's gone to the proteasome, it's gone. The anaphase promoting complex has to also now ubiquitinate and degrade a protein here that's keeping the enzyme separase from working. Separase separates your chromatids. That's all you need to know. Separase is inhibited by a protein called securin. It secures your chromosomes. Okay? So the mechanism for the anaphase promoting complex is securin is what gets ubiquitinated and degraded by the anaphase promoting complex. Remember, MPF is what activated this. So the anaphase promoting complex ubiquitinates securin, causes it to be degraded, and now that frees up separase, and separase comes and clips the cohesin. That is what is holding the sister chromatids together. Clip those, and now they can pull apart. In essence, that's what you go into anaphase. But now what we have to do is to link the activation of this process here with equal tension on the sister chromatids. And that's what the mitotic spindle checkpoint does. So here's the video again. And the thing to watch with the video is, for one thing, look how long it takes all these chromosomes to line up. You're going through all that trial and error process to get them captured and to get them moved. And there's this one pair of chromatids here that just can't get in there. And you notice that everybody else is waiting, 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 waiting. And that's because that one pair of sister chromatids is the only one that still has proteins that are phosphorylated by Aurora B. So as long as you've got one kinetochore protein that remains phosphorylated by Aurora B, the mitotic spindle checkpoint comes in and prevents the anaphase promoting complex from activating securin. So here's what we have. It's a pair of proteins up here called MAD1 and MAD2. MAD1 and MAD2 are the sensors. They can tell if there is a single phosphorylated kinetochore protein. As long as it's there, MAD1, MAD2 bind to the anaphase promoting complex and keep it from working. As soon as all of the kinetochore proteins are dephosphorylated, which means everybody was lined up in the middle, everybody's attached, everybody's under equal tension. At that point, and only at that point, are MAD1 and MAD2 turned off. And now the anaphase promoting complex goes to work. We ubiquitinate securin. It's taken out of the picture. And now separase can basically do it. So everything right here is waiting for that pair of sister chromatids to get in there. Boom. There you go. And the reason is, once you start anaphase, there's no going back. It's too late. So what this whole process is coming down to is protein phosphorylation keeps that from occurring until all the chromatids are under tension, they're dephosphorylated, and now we can go into anaphase. Cool. Okay. Here's what's happening in anaphase. Anaphase A, anaphase B. It's real simple. Anaphase A only is going to involve the kinetochore microtubules. Here's that close-up I was telling you guys. That's what the kinetochore looks like on a kinetochore microtubule. It's not, the chromosome is not actually out here at the very plus end. It's reached around, and it's wrapping around the kinetochore microtubule further ahead of it. Okay? So can you guys see that as we depolymerize this kinetochore microtubule, the chromosome itself doesn't let go. The chromosome itself is just going to scoot down towards the pole. And what's happening is that microtubule is depolymerizing right in back of it. The energy that's released by that depolymerization is pushing the chromosome down the length of that microtubule. Okay? It can't go back because there's no microtubule there for it to rebind to. So once we start this process of shrinkage, the kinetochore microtubules are shrinking at the plus end, and they're also shrinking down here in the centrosome. Okay? So the kinetochore microtubules are getting shorter. The net effect is the chromosomes move towards the pole. This is how Taxol works as a chemotherapeutic drug. Because what Taxol does is that Taxol, as you guys know, blocks microtubule depolymerization. So if you take cells and you treat them with Taxol, you cannot depolymerize the kinetochore microtubules. The chromosomes have no way of moving towards the poles. They can't move, the cell can't complete mitosis, and the cell will die. So that's the cytotoxic effect of Taxol in terms of being able to be an antimitotic. Okay? Anaphase B now is we're going to be driven by motors. And we've got two sets of motors that are going to be doing this. We've got these dyneins that are down here. So they're the ones that are attached to the plasma membrane. They're minus end motors, so they're reaching out and they're grabbing them towards the minus end of the astral microtubules and pulling them. So the poles are getting pulled back. So there's the dyneins. Kinesins are on these overlapping polar microtubules. Now you can see it right there. And what these bipolar kinesins doing is that as they walk towards the plus end, they're shoving the microtubule above them back in the opposite direction. And this one is also, since it's bipolar, as it walks towards this one, it's shoving that one back. So what's happening is the polar microtubules are going like this. Now, can you guys see there's a problem here? What's going to happen when the kinesins walk to the end of the microtubule? Can they go any further? No. So here's where the cell is going to recycle the kinetochore microtubules. So what happens to drive anaphase B is that all of this tubulin that's released from depolymerizing kinetochore microtubules gets recycled onto the polar microtubules. So at the same time the kinetochore microtubules get smaller and smaller, kinetochore microtubules get longer. And what that does is it keeps this region here of overlap, so this one will keep growing, that one keeps growing, and what you're doing is you're giving these kinesins more room to keep walking to shove the polar microtubules further apart. Okay? Can you inhibit anaphase B? Yes. You can inhibit anaphase B by blocking microtubule polymerization. And that involves using a drug, colchicine. So colchicine will prevent the polar microtubules from elongating, and that will block anaphase B. So if you use Taxol, you block anaphase A, because that prevents depolymerization. Colchicine blocks polymerization, so that blocks anaphase B. Okay, so we can stop the two processes by using two different drugs. Okay, here's the wrap. Here's the end. Now we get to cytokinesis. So anaphase A and B separated the chromosomes. Now we have to divide the cell. The cell, now we're switching over from microtubules to microfilaments, because the microfilaments are going to carry out cytokinesis. But they're going to do it from the inside. So when we're dividing a cell, the cell's plasma membrane here is actually being pulled in by contraction of microfilaments that form a ring. So the cell isn't being squeezed from the outside, it's being pulled, the plasma membranes are being pulled into each other from the inside. And microfilaments are doing that. And here's how it's going to work. The chromosomes are moving apart. We wind up with microtubules now reforming little fragments where the chromosomes used to be. They're called stem bodies. So what you're doing is as the chromosomes move out of the equator, we start growing new microtubules right there where they were. They send a signal to the plasma membrane around the periphery of where the stem bodies were. So in essence, it's a signal. You guys are used to thinking of signals coming from the outside of the cell to the inside. This is a signal that's going from the microtubules to the plasma membrane, and it's causing the microfilaments to polymerize, and we're making a ring of linear microfilaments like this. We're making a ring or a hoop that goes around right where the middle of the cell was. And in essence, what happens is this ring of microfilaments recruits the motor protein myosin, and in essence, it's just like muscle contraction. So the adjacent microfilaments slide past each other, and in essence, what we do is that we pull the cell in from the inside. So the plasma membrane is being pulled in and pinched off by the contractile force of microfilaments that are being told to do this from microtubules that are repolymerizing right here in the middle of the cell. Normally, that's symmetrical. Normally, you'll divide a cell in half right down the middle. But remember, that doesn't always have to be the case. Because when you looked at Saccharomyces cerevisiae, remember there that cell division is asymmetric. Okay? And that is the daughter cell that's going to bud off is much smaller than the original mother cell. So the key thing to understand is this can be either a symmetrical cellular division process or it can be asymmetrical. Okay? Okay, so that's a wrap on mitosis then. A lot of stuff happening. It's all happening at the same time. Okay? All right, so here's where we're going to start, the last section of the class. It's going to tie together signal transduction, cell division, mitosis, and most importantly, how do cells sense damage to DNA? So we're going to tie all of these together. It turns out that you can use cancer better than any other disease to integrate all of these processes and put them together. Okay? So what you guys are going to see is that as we go through cancer, the easiest way to understand it, and the first principle is, it's due to a failure of normal regulation during signal transduction processes. So that's going to explain why do these cells proliferate. And it's going to largely be due to the fact that molecules or proteins that are involved in signaling have undergone mutational events, and they're being deregulated. So these components are always sending a pro-proliferative signal. But the problem that we have to come to grips with is how did those mutations come about in the first place, and why were they selected for? So cancer's going to require us to go through understanding, well, what is the signaling pathway to begin with, and then also how is it going to be perturbed, okay? So we'll be starting on that on Wednesday. You guys already have the first unit uploaded, so we'll get moving there.
전화해?
아, 방금 제가 문자 했어요.
아 진짜 했어?
네,
오케이 오케이.
안 받았어요?
아 내가 꺼놔가지고.
아.
오케이, see ya.